Today, we are going to welcome Amanda Berry as our first speaker. She is coming, scientist from Los Alamos Labs. Today we wanted to bring in some of the cutting-edge research that's happening at Los Alamos Labs. I'm sure you'll hear a lot more about that from Amanda. In particular, how some of these biotechnologies are helping to perhaps combat some of the questions that came up yesterday. So full bios are on your program, so I won't go ahead and read that, so I'll just turn it over to Amanda. Thank you.
Hello. So today I'll be talking to you about tapping into biodiversity for biofuels, and specifically the biodiversity of algae. And so these are pictures of algae. So you can see they're all very different from one another. So up there on the top middle, that's a diatom. It's our most photogenic strain of algae. The diatoms are really nice and they refract really prettily because they have silica in their cell walls. Up here on the right top, this is Gaulderia. It's isolated from hot sulfur springs, so it's very tolerant to low pH and high temperature. Over here on your left, this is spirulina. So this is what some of you may eat in your food. And even though it's bigger than these other algae, it's actually a prokaryote. So it's actually more similar to a bacteria, whereas these other species are eukaryotes. So that's what we are. We're eukaryotes as well. But they're single-celled eukaryotes. And down here, this is Nanochloropsis. So you'll hear about that today as well. It's a brown algae, a stromenopyle. And then I don't know if you can see here through the chairs, but down here, this is chlorella, and I'll be talking about that today when I talk about my research. And just to show you how we actually culture these algae in the lab, we grow them on plates just like you would bacteria. So that's a plate up there of Chlorella sorokiniana. And so we actually streak it out on agar plates, and then we inoculate from that into flasks and then up to larger scale. So why algae? So for the United States, the interest in algae biofuels really was reinvigorated in 2005 with the Energy Policy Act. And this was expanded and extended by the ISA in 2007. And this dictates that by 2022, the United States will produce 36 billion gallons of renewable fuels. And those fuels will consist of, this is all cellulosic, so this comes from corn and sorghum, the blue here. And then this, which doesn't show up so well on the screen, but the white bluish bar, that comes from, it can come from waste or lignin or cellulose, also from grease waste. And then the red up there, that's algae, or what we call advanced biofuels. So those are fuels that can go into jet fuel, diesel, cars, a wide variety of places. And then this little bar, which you probably can't see very well, below the red bar is biomass-based diesel. So right now we're in 2018, so I don't know if you can see this with the chairs, but we're at about 26 billion gallons right now of renewable fuels. And actually most of that is cellulosics. So cellulosics, really we can boost it pretty high. So this slide, this is a slide that I actually stole directly from the Department of Energy, from the Bioenergy Technologies Office. This is a slide the director of that office presented in 2014, and it still applies now, just to show you the thinking at the Department of Energy for funding algae and other biofuels. In the United States, 13 million barrels, which is about 550 million gallons of fuels are required daily for the United States transportation sector. And about 80% of that is gasoline for motor vehicles. About 1.5 million barrels is for jets. And so most of this is for cars. And then some of that, the remaining, is for chemicals and products. And so the opportunity there for the United States is to replace these petroleum fuels with biomass fuels. And biomass is the leading renewable resource. As you can imagine, you grow up plants or you grow up algae, and you're able to do that over and over. And there's an estimate, the Department of Energy, the Bioenergy Technologies Office just released a report in 2016 that says there will be 1 billion tons of biomass, so this is plant biomass and algae biomass, in the United States that can provide for vehicles and aviation, make chemicals, and produce power for the grid. And they estimate that 30% of the United States petroleum usage could be completely replaced by terrestrial biomass by 2030.
Does that include natural gas?
Natural gas? No. Yeah. And so why algae? The benefit of algae is that it has a high productivity relative to plant feedstocks. So this is a map that was made in 2011, and this is using the best scenario. So this is saying if you had algae that was very productive at up to 25 grams per meter squared per day in your ponds, and you could see that it would use a lot less land than something like corn grain ethanol or a cellulosic ethanol. So it has a lot of potential. And this is if the pond doesn't crash. This is if it's year-round and it's growing well. It also is able to be grown on marginal lands. So unlike your plant feedstocks that have to be grown on fertile land that could also be used for food, algae has the benefit of being grown in the desert in shallow ponds or near coasts, near saltwater sources. It's also able to grow on saltwater or also waste. So there's a lot of projects looking at actually bringing in wastewater and just inoculating algae and getting the biomass that way. There's also efforts to clean up waterways with algae. So there's something called the algae turf scrubber, which is actually just a long trough, basically, of algae that's been inoculated on this trough, benthic algae that sticks to the trough, and then they run water cycling over it, and the algae removes any harmful things in that water, and then the water is recycled back through. And that's going on right now in the Salton Sea in California, if you're familiar with that area. Algae is also able to, it does photosynthesis just like plants, so it's able to recycle the carbon dioxide. And it has the potential to provide valuable co-products. So right now, the largest algae ponds that are economical in the world and in the United States are algae ponds that produce co-products, so that produce omega-3s, so Nanochloropsis can be used for omega-3 production. Spirulina ponds in Hawaii, that's where we get a lot of our food, supplements. Also astaxanthin, it's another pigment that's made from Haematococcus. So right now, that's where these big algae ponds are, and that's what's informing a lot of the research that's going on for biofuel production, because they've already addressed some of those challenges with their strains. Also, algae has the potential to make a wide range of biofuels, so including jet fuel, which is something that the cellulosics are not able to do. Let's see. Oh, and the estimate is that right now, algae could it could contribute to biofuel production by about 5 billion gallons per year. And down here, I'll show you these pictures in a little bit as well. This is actually in New Mexico. This is Sapphire Energy that's south of Las Cruces. And there's a nice plate again to remind you how it goes from that to that. So how do we actually get oil from algae? So when algae are under stress, they store all their carbon and they put it into these lipid bodies or oil bodies, and they actually just store it like fat pockets. So, and it's because they're under stress from either nutrient deprivation or temperature stress, salinity stress, heavy metal stress, pH stress. So any type of stress, they'll start storing those lipids. And this is just to show you a picture of that. You don't really need to understand all the components of these. Basically, the top panel, those are those single algae cells, cross sections of those cells. And they don't have very big oil bodies. And then when you apply a stress, you get these huge fat globules in the cells. And you can actually see that under a microscope. We can stain those oil bodies with different dyes so we can see the oil increasing in the cell. And then in an algae-based fuel process, you'd have your open algae ponds, and you'd have your algae making that oil in those cells. You would then harvest those cells, and that's a very energy-intensive process. So there's a lot of research going into how to make that more economically viable. Right now, using big centrifuges is just very energy-intensive, as you can imagine. So they've developed ways to filter the algae through membranes so that you actually harvest it that way. And then you can imagine you get fouling of those membranes, so there's a lot of engineering questions around that as well. And then you take that algae, and the nice thing about algae is that you can refine it in a normal refinery. So it's a fungible fuel, so it can be put into the existing infrastructure. And right now there's ways, the Department of Energy also funds ways to break open that algae and get those lipids out. Right now the favorite method is hydrothermal liquefaction, so it's just high pressure or high temperature to pop those algae open. And then you have your drop-in replacement then for jets or cars or trucks.
Can I ask one question?
Yeah.
Or do you want to wait?
Oh, no, go ahead.
Could you use the polymerization process to help? I used to work in Forest Product Labs at Berkeley,
Mhm.
but we used the gas, and we made like artificial blood,
Mhm.
super stronger. But I was wondering, we used a temperature pressure process
Mhm.
with the computer, and I was wondering if it can polymerize fibers, that amount of temperature and pressure, could that be used to extract the oil from the inside of the,
Right. Yeah, that's basically what that hydrothermal liquefaction process is doing.
oh, okay.
And then also, the nice thing about algae ponds is that you can imagine you're putting in carbon dioxide from sources that are emitting this carbon dioxide. So there's actually a lot of work right now going into, can you use flue gas to actually bubble that into algae ponds to get that CO2 utilized by the algae. And again, in these ponds, we're looking to make a lot of biomass, a lot of lipids, and also a lot of carbohydrates. So within the Bioenergy Technologies Office, there's an algae program dedicated specifically to algae biofuels. And they are what fund a lot of the work that we do in New Mexico. And their goal is to get to $3 per gallon gasoline equivalent by 2022. And so right now we're at about, in the best case scenario, at about 750 GGE of biofuel. And you can see right now in the last couple of years, because fossil fuel prices have dropped, a lot of the big algae industry that had a lot of momentum died away just because it wasn't economical. And also within these programs, they use a lot of techno-economic and life cycle analyses to see what's going to work. And that's really coupled to the research. And these are, again, just some pictures of different algae facilities. So over here, this is Arizona State University. They have a big algae program there. And again, this is in New Mexico. So this program has funded several projects in New Mexico. Funding from the Department of Energy goes back to the 70s with the Aquatic Species Program. And this was a long program that was started by Jimmy Carter to examine the production of energy using algae. And a lot of the initial work was done at this time, and that's what we're building off of now. And this was actually in Roswell. This is a test facility in 1987. And these were the first outdoor ponds that were built to test algae for biofuels specifically. And they actually looked at, I think the number was up to 3,000 different strains of algae at this location. And so again, they wanted to build this here in New Mexico where there wasn't land that would be used for crops, and it was in a dry climate, and they could actually see how the algae performed under those conditions. More recently, Sapphire Energy in Columbus, New Mexico was the largest algae biofuel facility in the United States. And it consisted of these large raceways, and it was started with about $50 million from the USDA Loan Guarantee Program and another $50 million from the Department of Energy. But in 2017, due to fossil fuel prices crashing, they basically closed their doors to biofuels. And now these ponds are still going, or a few of them are, with Chinese companies that are actually looking at co-products. And so what are the algae that we actually put in these ponds? I already showed you that picture of how different they all look. They're also very different in their biochemistry, their biology, and their genetic makeup. And so this is the eukaryotic tree of life, and we're way down here. We don't really matter. And algae, as you can see, are part of 4 of the 5 eukaryotic supergroups. So they're very distantly related to one another, these different groups. And the ones with stars here are just showing you which ones are being considered right now for biofuel production, or are big contenders due to their lipid content and how well they grow in the conditions that we're looking for. And you can see they're the red algae, which is that Gaulderia I showed you earlier. They're the brown algae, this Nanochloropsis, and the green algae over there on that tree. And then these little arrows here, those indicate endosymbiotic events, so when one cell engulfed another. And then down here, this Venn diagram is just showing you, these are the number of genes they actually have in common. So you can see there are many genes they have in common, but there's many genes that they don't, in the hundreds. And so when we look at these algae, we're trying to develop different tools to optimize them. And some ways we can do this is using genetic engineering or genetic tools. Some of these actually don't involve putting a gene into the organism, but they involve manipulating the organism's environment so that those gene changes occur. So there's directed evolution, and that's when you exert pressure on the algae. And you select for traits that you're looking for, so high temperature, low temperature, whatever environment you're going into. There's also actually engineering the algae with a heterologous gene, so that means a gene from another organism, and putting that into the algae. We can also use transcriptional regulators like promoters, repressors, transcriptional factors to alter the gene expression in that algae, to turn on genes that we want to activate and turn off genes that we don't. You'll hear a little bit more about transcriptional factors in Victoria's talk after mine, and she'll be talking about homeobox genes or Hox genes. There's also genome editing, so you may have heard of CRISPR-Cas. So that's actually where you're able to go in and specifically target genes and slice them out or put them in. Before CRISPR-Cas, we had things called zinc finger nucleases that did this, or TALENs, transcription activator like effector nucleases. And all of these are meant to do very precise cutting within the genome. And the nice thing about CRISPR-Cas is that you could conceivably insert it into the organism, slice out your gene, and then it wouldn't be present anymore, so you could get rid of it. So theoretically, that organism would no longer be a GMO because there wouldn't be a foreign piece of DNA in that organism. We're also looking at breeding. So some of these algae we know can mate, but not all of them. And we still don't know how to actually induce mating in some of them. Some of them, you actually induce it with certain wavelengths of light, so blue light. But we don't know how to do that in all of the strains. But you can imagine all the progress that's been made in plants due to mating and breeding. If we could harness that in algae, there would be a lot of potential there.
Can I ask one more?
Sure.
This is a really kind of dumb, basic question. but
That's okay.
Is that they grow it in water, right?
Uh-huh.
And then how deep, how, like,
Deep are the ponds?
how, yeah, like,
Mm-hmm.
in other words, if it was 1 foot versus 2 feet, I'm just making this up.
Mhm.
Does, and the sunlight.
For the light penetration.
sun comes down,
Right,
does-
right.
does it matter?
So these raceways, yeah, I didn't mention that, but they're very shallow. So they're only about 20 centimeters maximum deep. And then there's also paddle wheels that paddle that algae around to keep it mixed and to keep the cells seeing light. Yeah. Yeah, good question. And so again, this is the idea that we're trying to take algae in a population and make a super algae. So that's really the goal of these processes. He's so happy. So we also use omics approaches in bioinformatics. So you probably heard genomics. So that's the study of the genomes of these strains. So understanding all the genes that are present and looking at how those may relate to biochemical pathways and how we can manipulate those genes. There's also transcriptomics. So this is looking at the RNA in the organism. And that's, you go from DNA to RNA to protein. And I'll show you that on the next slide too, just to remind you. And that's the message that comes from that DNA. And so we can look at that to see when these genes are turned on or off, or when they're being made into protein potentially. And then that goes hand in hand with proteomics, where we actually look at the protein concentration in that cell to see when these proteins are expressed. So proteins are also enzymes, enzymes are proteins, and so we can see when those go up or down. And so this just shows you that because I'm focusing on eukaryotes have a nucleus, and so you have your DNA in the nucleus, and that codes for your RNA, and then your RNA gets coded into protein. So DNA and RNA are both composed of just 4 nucleic acids, and then your proteins have the potential of 20 amino acids, so more variability. So we also use a lot in algae metabolomics, lipidomics, and glycomics. So this is all looking at small compounds within the cell. So metabolomics is looking at any small molecular weight compounds that are involved in growth or the function of the cell. The lipidomics is looking at the lipids, so that's looking at those oil bodies, looking at which types of lipids it has, because that informs which kinds of fuel it can make. Also, we look at the glycome, which is the collection of all the sugars or the carbohydrates within a cell. And I'll talk a little bit more about that later. And then overall, we look at the phenomics. This is looking at the phenotype of the cells. This is how it behaves in different environments. And that's an overall understanding of what's going on in the cell. And so again, I'm looking at these diverse algae, trying to apply these tools to strains that are very unrelated to one another is very difficult. So when we're trying to do genetic engineering, we have to develop toolboxes that are specific for each of these strains. So even within the same species, there can be strains within that species that actually differ enough genomically that we need to develop entirely new genetic toolboxes. And the same thing when we try to apply different tools like lipidomics or metabolomics. All of these strains have different cell walls, so it's difficult to penetrate them in some cases to actually get at these compounds. So each of them is very challenging. And so So some have taken the approach of actually just tapping into the natural biodiversity. So there's enough strains out there. Can we select for the traits that we want by just investigating enough algae that we can then get some target strains that have high lipid, that grow very well, without having to manipulate them. And so this is a project that I just wanted to highlight that was funded by the Department of Energy, called the National Alliance for Advanced Biofuels and Bioproducts. And this was 39 partner institutions, so Los Alamos National Lab was one of these. And just one aspect of this program was actually doing this bioprospecting. So they actually went out and sampled algae from different locations all around the United States in different types of ponds, saltwater, freshwater, brackish water. And they started with 2,200 independent strains of algae, which were present in 400 samples. So multiple algae are present in each of those samples. And then they isolated them by single colonies on a plate and screened them in large flask screens, and then characterized them. And from this study, out of 2,200 independent samples, they found about 30 strains that they thought were worthy of further outdoor testing, and those were deposited into a culture collection that we have at the University of Texas, and so that's where we get most of our strains. They're all kept there. One of the challenges with algae, unlike prokaryotes like bacteria, is that they're difficult to freeze and store, and so a lot of strains are lost because you just have to keep culturing them constantly. And you can imagine in the lab with that directed evolution, you're going to be changing those strains over time by just keeping them in a lab environment for years. And under NAB, that's what we call it, NAB, I just wanted to show you this. These are all, each one of these represents a different strain of algae that was examined. And these little pie charts represent the lipid profile. And so they all differ from one another, so those composition of lipids. And that's very important for when we want to consider downstream what the fuel is going to look like. So these are all different, so really for each algae you can't make assumptions. You have to really do all of these tests. So at Los Alamos National Lab, one of the things that came out of NAB was the development of this environmental photobioreactor. So that's one of these little machines here. Actually, I think I have, yeah. There's a close-up right here. It's as big as me. So they're about as big as a coffee maker. These hold about 500 milliliters of liquid, so they're the same depth as a raceway pond. So you can see they're pretty shallow. And they have bubbling coming in from the bottom and a stir bar to mix them and light coming from the top. And then you have these fans on the side to heat and cool. And so these were a big deal for the field of algae biofuels because there's really not a good way to predict that what you're getting in the lab is going to work when you go outdoors to these very large raceways. So you can imagine a flask with light coming in from all sides is going to be very different. And that environment is very different than what you're seeing outdoors. So the goal of these was to try and provide that environmental mimic so that we could do those tests in the lab and do them in replicate because we wanted statistical significance, which is also difficult to do outdoors in these large ponds. And so these provided a way to do that. They're also enclosed, so if we create genetically modified organisms, we're able to do those in these as well and compare them to the wild type or the original strain. And so they're very powerful in that way. They actually underperform for what we see outdoors, so they're not perfect, but they get us a little bit closer. So Los Alamos National Lab and the New Mexico Consortium, where they're housed, we have about 33 of these right now. And I think there might be a tour to the NMC on Sunday. So you'll see these if you go on that tour. And so again, the idea is that we're going from these plates that we're streaking out, then into flasks, and then into these EPBRs. And then we take them outdoors. This is the facility at Arizona State University, who we work with. They have a test bed facility that's funded by the Department of Energy to test all these strains at one location in replicate in triplicate so that we're able to get meaningful data. Also at Los Alamos National Lab, we have genome sciences. I would say in the United States, Los Alamos National Lab is the foremost institution for algae genomic sequencing and transcriptomic sequencing. They're also getting into proteomics now and becoming a lead institution for that. So people come to us with our strains. Under NAB, several genomes were sequenced at Los Alamos. And then we're able to use that sequence to get the answers that we're looking for. Sure.
Okay.
Yeah.
I was like reading this article that my friend gave me in Smithsonian, and it was talking about like some studies done, I think it was 2008, and they found that they were looking at a million cells, they weren't looking at algae, but they found out there was like 620 million like proteins. So I was just wondering, like the 33 out of the 2,200 algae, but even though they're all distinct and everything, you gotta test them out in the wild, I was just curious if there was any commonality in terms of like how much protein or what types of protein are
Right,
in that,
right.
in that algae.
Right. I mean, they definitely do have similarities in their biochemical pathways and the proteins that are responsible in those pathways. But yeah, nobody's actually measured the total protein in the cell for any of those algae as far as I'm aware. They all differ in genome size a lot. And they differ in their biochemistry, so the pathways that they actually have. So some may express proteins that others don't. But yeah, examining that has not been done for all of those strains.
The surprising thing was they discovered that this is human cells now, that there were far less genes than they realized, and it was protein was like taking up all this space. And
Right.
I know they're studying at Sampling Institute, and aren't they like studying ribosomes?
Probably.
Right?
Yeah.
Those are the ones that thread the little RNAs, right, and make the little.
Right, right. Yep, yep.
Oh, thanks.
Yeah. Yeah, biochemistry is really cool. I like it a lot. And so this is at Sandia National Lab, so just to show you, it's not just Los Alamos, so Sandia and Albuquerque, they also do algae, and we work a lot with them on different projects under the Department of Energy. And they just built recently, this is, I think the ribbon cutting was just in 2016, this algae raceway testing facility. And this was specifically to test algae crashes. So as you can imagine, you're outdoors and you have this large monoculture of algae, and you can get predators in there. So there's rotifers that actually eat the algae. There's other algae that could outcompete them. There's also, you can get bacterial infestations. And so they test that in this facility where they're able to enclose these ponds and have these lights on the top to be able to mimic an outdoor raceway. And then they have the ability to actually hyperspectrally monitor the cultures to actually see when the algae start to change, to try and predict when these crashes will occur or when they are occurring. Because you can imagine, just like if you have a plant crop, if you're going to get a pest or some kind of crop death, you want to try to detect that before it actually happens so that you can apply some sort of remedy and hopefully prevent it. Or you could empty your whole pond and re-inoculate it too. So this is a project that's funded right now. This just started last year, and this is includes Los Alamos National Lab, Sandia National Lab, and 2 other national labs, Pacific PNNL, and NREL up in Colorado. And this is specifically trying to tap into that biodiversity. So a lot like what NAB did, but now focused in on 30 strains, and really taking those strains and scaling them up and doing different analyses to determine how stable they are outdoors, how much lipid they produce, and which temperatures they can survive in. And this is using media that they know doesn't take a lot of nutrients, it doesn't cost too much, so that it will hopefully be able to be used outdoors. And so there's no genetic engineering in this effort. And so what are they looking for? So the traits of the superalgae. So you want temperature tolerance, you want tolerance to high temperature and very low temperature like we get in the desert. You also want low nutrient requirements, so not a lot of nitrogen or phosphorus or anything being put into that pond that costs money. Salt tolerance is also very good because you don't want to be using fresh water that could be used for drinking water, using brackish water either from the ground or salt water from the ocean is preferable. Having rapid growth in those conditions, and that means being resistant to photosaturation. So when you have high light, just like plants, you'll get bleaching, photobleaching of the chlorophyll in those algae, and they'll start to yellow, and some will start to die, but some are more tolerant of those high light intensities. You also want a tolerance to oxygen saturation, so they're still doing photosynthesis and still active. And you want them to be able to efficiently capture that CO2 and turn that into lipids or into growth. You also want stability over time with fluctuating conditions, so that's when it rains, you don't want the algae to crash out. Also, a big problem they had at Sapphire Energy was actually tumbleweeds. So these tumbleweeds in the desert would just collect in the raceways. You can imagine, you see them on the side of the road on the way to Albuquerque, but just, tons of tumbleweeds that they would have to clean out of the ponds, and they would just introduce different organisms. They also had a lot of difficulty with birds just landing on the ponds. They had a lot of interesting stories about different wildlife that would wander in and use the ponds in different ways. So those are all challenges you have to consider. Also, the resistance to predators and competitors, like I was talking about earlier, you don't want your pond crashing out. So you want algae that are very hardy, very stable, able to outcompete anything else in the environment. And again, you want that high oil production. And hopefully you can do that without actually doing starvation of nutrients or that loss of growth, because when you take away nutrients, it stops growing and it makes more lipid. But that also takes a lot of time for it to grow up, get rid of those nutrients, and then starve and make oil. So it'd be nice if it just made oil all the time. So again, this is the algae-based fuel process. So another characteristic that we can think about is that at this point of cultivation, we already know that we could get a lot more biomass in some strains if we actually added sugar to the ponds, because that's another carbon source. But of course, you're going to get rampant contamination. Sugar is very expensive, and so there's a lot of things that are against that. So one of the questions that I ask in my research is, could we find strains of algae that actually utilize biomass? So we know there's going to be this billion ton of biomass. We also have a lot of biomass left over from after food is harvested from the plant. So you have something called corn stover, so that's the actual stalk of the corn that's left over. And there's a lot of efforts to actually collect that biomass and use it in different ways. So one of the questions was, can we find algae that are able to eat that biomass and actually boost their energy that way? Along with CO2. So it's called mixotrophic growth. So they're able to do photosynthesis and they're able to use organic carbon to make more lipids. And so I actually found this strain that came out of NAB called Oxinochlorella prothocoides. And this is a high lipid producing green algae. Unfortunately, it grows on fresh water or water that isn't too salty. But it was known to like sugar, so it could grow on glucose and different types of sugars. So we decided to test it on cellulose alone, which is a component of plants, just to see if it had any activity. And so this is called a Congo red stain. So you streak your algae on a plate that contains cellulose, and this red stain binds to the cellulose. And so if you have clearing of that cellulose due to the organism breaking it down, you'll get a clearing in that red stain. And so what we can see with the Oxinochlorella, hopefully you can see it, is that you get this huge clearing, which was very surprising for us. So we only know of one other algae that can, at this time, that could utilize cellulose, and it was Chlamydomonas reinhardtii, which is a lab strain of algae that's been used for forever in the lab. It would never go outdoors because it isn't hardy enough. But we knew it could use cellulose, and it just cleared a little bit, like just right where the cells were. Whereas oxynochlorella seemed to actually be putting something out of the cells that was able to degrade the cellulose around it. So it's actually secreting some enzyme that breaks down that cellulose. So we wanted to characterize this. And we also wanted to test the growth of this algae now on different plants, because that's what we're most interested in. We don't want to add pure cellulose to the pond. Other things can use pure cellulose. The nice thing about raw plants is that not many organisms can actually break that down and use that as an energy source, so you won't get as much contamination as if you use a simpler substrate. So we wanted to test the growth on all of these different plant residues. This is raw biomass that we get from Idaho National Lab. So they have a feedstock library that's funded by the Department of Energy to test these different types of biomass that are used for biofuels. And so we have switchgrass, eucalyptus, we have corn stover, silver birch is another one, and there's also pine. And what was interesting is that we did this with switchgrass, which was the one it liked the most. And we always do this in triplicate in flasks. That's always very important in science, make sure they do it in triplicate. And what we found is that when we added switchgrass to the flask, this red line is much bigger than the blue line, and we actually saw an increase in growth. So it's actually able to use that switchgrass for energy and growth. And also, interestingly, we saw that the lipids increased when we had the switchgrass present. So it's actually taking that organic carbon and storing it as lipids. So then we wanted to ask, which part of this switchgrass is actually being eaten by the algae or being used by the algae? And so that's when we do these glycomics. So you don't have to understand this picture at all. It's just really, really pretty. And this is an art museum. And so I love showing it just because it's neat. So each of these bands corresponds to a different type of sugar in the cell. And the color of those bands correlates with the intensity or the quantity of that sugar in the cell. And so now this is looking at the plant residue after algae treatment, basically. So we're adding the algae in the culture, or we have plant alone in the media for the same amount of time. And then we're comparing what happens to those plant sugars. And what we see, and again, you don't have to, this is just for people who really like chemical structures. This is hemicellulose. And what we found was that the algae was eating the hemicellulose, so it was eating that fraction of the plant. And that's part of the cell wall, along with lignin, which this organism does not seem to eat. It prefers the cellulose fraction. And those are also known as xylans. And so then we took that xylan fraction alone just to see which part of the xylans it preferred. I'm just going to show all this. And I don't know if you can tell, but up there we have without algae is in that green box without a plus sign in it. And then when you have the algae present, it's got a plus sign. And so you can see that column there has a lot fewer bright bands than the one without the algae. And so those bands are disappearing when the algae is there, and that's breaking down those xyloglucans. And interestingly, the cellulose in the wood is cross-linked by these xyloglucans. And so this told us that the enzyme that's being secreted by this algae is probably a xyloglucanase, because with enzymes you just add an ace at the end, and that's all it is. And that's also known, it belongs to a family of proteins called glycosyl hydrolases, which include cellulases, which break down cellulose. So we wanted to look at those glycosyl hydrolase families. What's also interesting about this is that we went back to the culture collection and determined when this algae had been discovered previously. So we knew it was a NAB candidate, but we also learned that in the 1800s, Oxynochlorella protothecoides was isolated in Germany from a tree. So it was actually part of some wood on a tree, and so it makes sense that it would be able to create a niche in that environment by breaking down some of that carbon. So then we did transcriptomics. So again, this is looking at the RNA, so looking at what's being made into message in the cell. And you, again, don't need to understand all of this. This is a tree. So these are all the glycosyl hydrolase sequences present in this algae. And it has a lot of them. So this is a lot. So generally, Nanochloropsis, for example, that actually has cellulose in the cell wall. So we would expect it to have cellulases and different glycosyl hydrolases to recycle that cell wall and make new cell wall. And that has about 6. And so this is a lot. So this does definitely tell us that this organism is involved in this degradation, doing something with the substrate. And what we found were that these 5 that are in boxes here, the green ones significantly, were upregulated or increased when the plants were present, when that substrate was present for the algae. So we think it's one of these enzymes that's involved in that degradation. So we can look at them more closely. So we call them 5 and 9 because those are the families of glycosyl hydrolases that are known to include cellulases. And we can do these sequence alignments with other proteins that are known to have this activity from other species of bacteria, for example, in the gut. They also break down cellulose because we eat it. And we can look for specific residues or amino acids in that sequence. So again, there's 20 different amino acids that can go into proteins, and we just look at specific ones to see if it might have this activity because they're involved in that activity. And again, this is just for people who really like chemistry. And we can do that for both families and see that they have the potential to have that activity. So then we can follow this up with proteomics. So this is actually looking at the suite of proteins in the cell and how they change when plants are present. And this is all done actually outside of Los Alamos. This is done at Kendrick Labs in Madison at the University of Wisconsin-Madison. And each one of these spots in these pictures is a different protein. And we can actually run all these proteins on a gel. So they break the cell open. They run all these proteins out so they get these individual spots, and then they can cut out these spots, like physically cut them out, and then sequence them and determine their protein sequence. And we do this for plant alone, we do this for algae alone, and then we do it with the algae plus the plant, and then we can subtract these. And this is what a subtraction plot looks like. And then we can cut out the spots that are different. And when we do this, we just sequenced 8 of them, and we compared it to the transcriptomics. Surprisingly, we didn't find any glycosyl hydrolases. But we did find that the protein that was most expressed was unknown. And it actually has the potential to bind a sugar when we compare it to other proteins out there. So this gives us another candidate that we can use to try and determine if it has this activity. And so really you can see we get different candidates from different experiments. The other 7 spots were all involved in glycolysis, which is using this carbon in the cell. So all those pathways were upregulated, which shows us that the carbon's actually being used in the cell. And this is just a model that we can make. So with different programs, modeling programs on the internet, you can find these and model your protein structure. And they look pretty. And so this is the project that I'm working on now. This is funded by an Early Career Research Award at Los Alamos National Lab. And this is just taking algae, testing it on different types of biomass in the EPBRs, and then down selecting and scaling that up to determine if it behaves the same way when we go into more outdoor-like conditions. And we're also looking at engineering a saltwater stream. So once we determine the proteins that are involved in Oxina chlorella protothicoides, we can take those proteins, we can put them into other strains of algae that can grow in different environments, such as saltwater, and hopefully engineer them to have that same activity. We're also doing some bioprospecting ourselves for other organisms that utilize different cellulose substrates. These strains here were actually isolated from pulp mill ponds up in Canada, and so it makes sense that they would be able to degrade cellulose. And so at Los Alamos National Lab, we were given these strains to sequence and test them, and we were able to determine what they were. They're both chlorella species, so it seems that these Oxena chlorella is similar to chlorella. These green algae, they really like these cellulose substrates. Chlamydomonas also is a green algae. And so I just want to end just reminding you that these are all very diverse organisms. What you say about one algae isn't the same for another. They all differ very much biochemically and biologically. And so really, I think it's neat. And there's just so much that we can learn from them still. And so this is the New Mexico Consortium. So if you go on the tour tomorrow, this is what you'll see. It's a really nice new building. And then these are my students up here who I had. Alondra Villalba came in through a NSF program. And she was in my lab last summer. And then Brian Vogler, he's a grad student at Colorado School of Mines. We have a great program at Los Alamos for bringing in undergrads and graduate students from other institutions for the summer, which is really nice. And then right now I have Jenna Schambach. She's just got her master's on the East Coast and then is coming to work for the lab for a couple of years before she decides what she wants to do. So that's it. Thank you.
I wanted to just bring something up. The presentation yesterday by Chris Kempis went through the basic cell processes from DNA, messenger RNA using the ribosome. So we're going to put that online at bioculturasantafe.com in about 2 weeks. So if you want to check that out, it was really fascinating. I have a really dumb question.
Oh, no, no dumb questions.
So the algal life cycle,
Mm-hmm.
isn't there a part of the life cycle where they die and let off a whole bunch of methane and greenhouse gas, and how is that dealt with? And if that's the case, which maybe I'm mistaken on that, how was that managed?
I don't know about that.
You can just keep them alive?
Well, I mean, it depends on the algae, so if they mate or not. But yeah, so there is cell turnover. So you'll see, what's interesting about algae is you'll see them grow during the day when they're growing phototrophically, so just on the light. And then at night, you'll get this little dip. And then they'll grow again, and then you'll get a little dip. But as long as you keep providing those nutrients, they'll keep growing. But yeah, they do sort of die off. But it's generally thought that die-off, that those nutrients, when they explode their bodies, are used by the other algae around them. So I haven't heard about any methane off-gassing or anything from ponds, but I'll super look into that. Yeah, what's also interesting is some algae actually shed parts of themselves as they grow. So Nanochloropsis, one of the strains of Nanochloropsis actually sheds its cell wall, so you get these cellulose cell wall pieces that actually fall into the ponds, which is interesting. Sure. Yeah.
So from what I understand, there's a real advantage to the open ponds doing this in desert climates, dry and a lot of sunlight.
Yes.
But are there possibilities of these open ponds in other bioregions? And
Yes.
is there like a map of the U.S. of
Yes.
where it's being done now and
Yes.
suggestions of how we see this
Yes.
grow
Yes.
in all sorts of places?
Yes, and a lot of that work's being done at Pacific Northwest National Lab, so actually mapping different regions that would be best for algae productivity. As you can imagine, Florida, the panhandle is like an optimum place because it's humid and hot and light. And also, the largest ponds right now for algae are in Hawaii. So that's where the spirulina's made in the United States. And they're able to pump in the seawater and actually filter the seawater because they don't want any contaminating organisms and grow it in those large raceways. There's also ponds in Texas that are being used right now for omega-3s, so large nanochloropsis ponds. And they actually put sort of like a cheesecloth over the top of the ponds because it's so bright and you get that photoinhibition, so they don't want that much light because you saturate all your algae pigments right away. So yes, there are definitely maps of these ponds. There's also closed systems, so there's closed photobioreactor systems when they're trying to make specific products or use GMO algae. You need to enclose them in they're these clear bags, basically just like plastic bags that are hung on these racks. And that was going on in Florida at a company called Alginol. And they had a cyanobacteria, actually. So spirulina is a cyanobacteria, and there are other strains of cyanobacteria that people are using.
So the more northern climates, it's not.
No. Canada does have an effort to make algae biofuels because they're just so darn good. So they have their own government-funded algae program where they're looking at strains that can grow in colder climates. So does Scandinavian countries. They also have efforts to do that. Denmark has an algae program. So they're looking at trying to grow strains either in indoor systems or artificial light systems, which can be really expensive and use a lot of energy, or have these cold adapted strains and just grow them in the summers and then just cut them off in the winters. Mm-hmm. Yeah.
Are there other collaborations with artists?
With artists. Well, the diatoms, they have a lot of potential. I mean, yeah, sure. I mean, definitely. I mean,
Aware of artists who are working with algae?
no.
There's a bunch of them.
Really? Oh, like dinoflagellates or something that lights up? Or because I know I've seen, there's products that have algae in them, like those little dino things. I don't know if you guys have seen those that light up. But yeah, that's interesting. I should look into that more. Yeah. Oh,
We should probably.
okay.
Next one. Thank
Okay.
you so much.
You're welcome.